Hi, everyone. Welcome to Foresight's Biotech and Health Extension Group, sponsored by 100 Plus Capital. Today, I'm very excited to have Varun Dwaraka here, who is a Foresight Fellow this year. So thank you so much. And I'm really excited to have you here. I think you'll be discussing a topic that definitely has been on everyone's minds for quite some time. And I'm really excited for you to share some new results of what you guys have been working on. In particular, you'll be talking about how we could possibly move beyond chronological age-based biological clocks. So lots to say on this front, in particular towards DNA methylation clocks. But I think you can do that much better than I can. So without further ado, I'll leave the stage up to you and I'll be sharing more info about you in the chat. Thanks everyone for joining. After about 30 minutes or so of presentation, perhaps a bit longer, I'll be moderating questions and taking them from the chat. So if you want to make sure your question gets asked, already put it in the chat. And otherwise, we also do just raise hand afterwards. Okay, Varun, the stage is yours. Thanks a lot for joining.
Thank you, Allison. And also, just as a FYI, I do have a lot of slides. So the background, I will try to go through a little bit quicker, just so we can get to the actual good stuff. Thank you again for allowing me this platform and opportunity to kind of discuss something that not only me, but like the rest of us at True Diagnostic are really excited about. And it's this topic of moving beyond chronological age-based biological clocks. And kind of, maybe problem is a strong word, but looking at these, some of the things that we can improve on and some of the recent solutions for DNA methylation clocks, including a solution that we have developed here in-house at True Diagnostic with collaborators from other institutions. And so with this said, I really need to say that this was a concerted effort with a lot of us at True Diagnostic with Dr. Jessica Lasky-Sue's group. And while the entire lab was instrumental in getting this developed, I do have to say to Qingwen Chen, who hopefully is on this call, thank you very much. So I wanted to give out the roses before we started to go in. And so with that said, I do want to explain who we are as True Diagnostic. Our mission, so we are an epigenetic company and we specialize in epigenetic testing, but our mission is to improve people's lives by arming patients and their physicians with information so that allows them to make right decisions at the right time, especially looking at the fluid epigenome. And so we were founded in 2020 for the purpose of leading and understanding and interpreting epigenetic data. And so we have a CLIA certified laboratory here in Lexington, Kentucky, which is about 20,000 square feet. And we've worked with a lot of academic collaborators, especially from 20 plus institutions. We've been able to publish, thankfully, on a lot of our collaborations, and we hold 10 plus more patents. And it's through these extensive relationships in clinical testing markets, research, and academic collaborations that we've really solidified ourselves as a leader in the epigenetic methylation lab services space, bioinformatic processing, and data interpretation. And it's through all this that we are able to do a lot of research and development, especially into epigenetic predictors and epigenetic clocks. And so with this said, I do want to go into a primer of DNA methylation diagnostics. I will go through this a little bit more quickly. Hopefully the community is able to understand this area very well, but I do think that having a primer allows us to have a better understanding of where we're going towards in our own research. And so when we consider epigenetic methylation, epigenetics can constitute DNA methylation, also histone modifications, and other facets of epigenetics. But specifically, what we're looking at is DNA methylation itself. And so epigenetic clocks are using the presence and absence of these biochemical marks at specific CpG sites to predict that epigenetic age. And I don't think I have to belabor this point, but this epigenetic age or biological age is different than your chronological age because it's based on the biology rather than that date of birth. And so the platform that we're using is typically the EPIC 850K array, but we're essentially quantifying these locations, the methylation at these locations by sequencing methods, and we use the beta values, or these values of relative methylation, on a continuous scale, and those are those values that are essentially being measured and being able to translate over to an epigenetic age. And so you might be wondering, why is it that you're focusing on DNA methylation? We've known that the effect that DNA methylation changes due to aging since the 1960s. And since then, we've uncovered millions of the 28 million CpG sites that change with age. And I do have to give my respects to Steve Horvath and also previously to Brocklin et al., the individuals that were associated with that paper, because those were really the two initial papers that showed the significant association of DNA methylation sites to age and really showing it as a highly predictive marker of age. And so with that, a lot of those earlier clocks were really developed using chronological age. And so again, training to that date of birth. And this allowed for that initial predictor to be a very forensic type age calculation. But in more recent years, instead of training to, let's say, chronological age, what newer models or newer age clocks have been able to do is actually be trained to biological age metrics. And so we've been able to do this and to integrate a lot of the current metrics that you take at your clinical lab values at the clinic and be able to convert that into a more biologically informed epigenetic clock. And so this has really led to various epigenetic clocks over the years. And just to give a taste of these clocks, and I will use these terms first generation, which essentially means trained to chronological age itself, or second generation, which is trained using aging phenotypes or those biological age measures. Just to give a taste of the expansive nature of these clocks, this figure, while it is a little convoluted, it kind of explains that we have many of these epigenetic clocks that are developed using both chronological age as a training model, but also biological variables as another training model as well. And so some of them include what I imagine that this group already knows: these Horvath clocks, the Horvath clock, the Hannum clock, the Zhang clock, and then also the Phenobio Age clock by Morgan Levine and her lab out of Yale, the Grimm Age clock, and then also the telomere clock. And really, I do want to say that a lot of where we're trying to move towards is kind of harnessing more of that biological age calculation, which I personally think that Grimm Age does really well on. Essentially, what Grimm Age is a clock that was developed using DNA methylation to predict various metrics or surrogate markers that we have already known influence or that are predictive of your biological age. And using that, we were able to regress that onto time till death and be able to identify predictive markers that allow you to quantify this Grimm Age value, which has been shown to show significant associations to typical disease onsets, such as cancer and cardiovascular disease and so on and so forth. And with that said, from Grimm Age, we've been able to go towards more of a clock that was developed through longitudinal clinical outputs. And so this clock called Dunedin pace represents what we consider a third generation clock, which is using aging phenotypes and measurements and produces an instantaneous rate of aging. This rate of aging is influenced a little bit based on your previous history, but it captures more of that instantaneous signal itself. And so Dunedin pace represents a huge breakthrough in that regard because it followed a cohort of 1,037 individuals from birth to 45 years of age, collected a whole slew of system covariates, so health covariates, ranging from cardiovascular metrics, metabolic metrics, renal metrics, immune metrics, dental, etc. And then using all of this information, they were able to generate one single pace of aging score just using these clinical values. And then using that pace of aging score, they were able to regress that onto methylation data itself of the same individuals and identify those predictive biomarkers that were highly correlated to these systems. But then that overall pace of aging showed also significant associations to disease onset and other disease metrics as well. So with that said, now I hope that, especially going back to this figure, I think it is apparent that there are a lot of clocks that are coming out in recent days. And so one question is, how do these clocks compare? And this preprint, I think now published by Morgan Levine's lab, actually did a very, very wonderful job of really cross-comparing a lot of these clocks to see what are similar, what are different, and really basing this off of this whole idea of these first three generations. And so what they found was that each clock may represent different CpG sites, so those DNA methylation biomarkers. However, generations generally share some similar modules. So for example, those first generation clocks share similar modules, and these modules are color coded so that that comparison can be made a little bit easier, whereas the second generations do as well. But between the first and second generation, that's really where you're starting to see deviations. For example, we spent a little bit of time talking about Grimm Age and Dunedin pace, or Dunedin POAM, which was the previous version. you could see that Grim age listed here and Dunine POAM generally share the same colors and also the same amount of those colors between each other. Those colors represent those CPGs and the biological processes that those CPGs are specifically associated with. DNA methylation, the DNA methylation telomere estimator, is also similar to Grim age and Dunine PACE, but then really have an enrichment of those orange modules as well. But really, out of all of this, the real takeaway is that even though there are similarities, each clock is still capturing something different, which is inherent to that training process. And so now once we start to talk about those clocks, really when we start to think about them in the context of how we can use them for patients or clinicians, really predicting the age is great, but then what does that mean in terms of the clinical output? So this is where we have to start to consider epigenetic age clocks and their association to disease and interventions. And so this review by Bergesma and Rogeva, which was published in 2020, Table 1 did a very good job of really summarizing a lot of these current clocks of that time and really looking at those reported associations. So you'll start to see that even if you're a generation 1 clock, which was again trained to chronological age, or a generation 2 clock or 3 clock, which was developed based off of those biological and clinical outputs, you start to see that they still have significant associations to, for example, the Horvath clock showing significant association to cancer, cognitive function, Parkinson's disease, and all-cause mortality. Whereas, let's say the Levine clock not only replicates some of those, but also increases that to cardiovascular disease, coronary heart disease, dementia, etc. And so each algorithm shows statistical significance to various disease outcomes. And this is not only limited to the the second and third generation clocks, but also the first generation clocks as well. But this really goes to show that as you start to impart more information into these clocks, these associations start to increase as well, especially when we consider the comparison between the Horvath clock to the Levine clock. And lastly, or next up, this paper in the Dunine PACE paper in one of the supplements, I think Daniel Belsky and the rest of the authors did a great job kind of comparing the Dunine PACE clock versus the Horvath, Hannum, Pheno age, and Grim age. And what I hope you can appreciate from this table is that the significant associations to different types of disease, such as cardiovascular disease, stroke, and even just all-cause mortality, are significantly shared. Those associations are significantly shared between the Dunine PACE and Grim age clock to each of these diseases. But that's where it starts to falter when we start to consider those previous clocks that were developed really on those chronological ages. And so ultimately, this going back to that idea of, in order to make this a little bit more appropriate for clinical and patient use, there needs to be an integration of these other metrics in a case of clinical metrics, protein metrics, and other metrics that really relate over to health as well. And this kind of comes to this next slide where, Dunine PACE and Grim age show significant associations to disease. And so what part of that training is allowing for this? And hopefully it's easier to see it in these two graphics, but really it's that inclusion of these different biomarkers that we've already known to be clinically relevant to that overall health and aging process, that including that information seems to improve upon the algorithms themselves and produce a clock that is much more clinically reliable and clinically informative to the end user. And so by integrating additional information than just the epigenetics, including clinical and protein values, seem to hopefully improve that. And so the question, or I guess the statement that really needs to be tested, is integrating multiple facets of data into a composite score. Does that translate to a better biological age prediction? And this kind of makes sense because if you consider what aging is, it's a very, very complex process, which not only takes into consideration just the degradation of cells and other biological processes, but genomic instability, telomere attrition, stem cell exhaustion, other processes like that. So being able to take into consideration all these other various pieces of data may allow you to have a better understanding of that aging process. And so that was where this approach that we tried here at True Diagnostic really came into play, is how do you integrate multiple layers of that aging biology? And one of the things that really came up to that really showed us, or seemed to make sense to us, was this idea of multiomics. And so, especially with the advent of next generation sequencing and the availability of different computational biology tools, really looking at all proteins, transcripts, epigenetics, genomics, metabolite data, and also just EMR data. How can we take all that information and condense it into one single piece of composite information, really? And so that was kind of our approach. And so we've been able to develop this composite score that we are calling OMIC MH, where we worked with collaborators from Harvard University, specifically the Mass General Brigham's Biobank. And we were able to allocate the proteomic data, clinical data, metabolite data, and methylation data as a way to really create this one tool. And so just for those of you who are interested in this biobank, essentially we developed a data set with matched multiomics, and we call this the OMIC Age Biobank, which serves about a sample size of 5,000 individuals that were collected from the biobank itself. This included clinical lab and phenotype values, metabolomics that were sequenced using the Metabolon platform, the untargeted proteomics, which was sequenced using the Seer proteograph method, and then also mass spec, epigenetics, which we used using the EPIC 850K array, and then also genetics as well. However, I will be talking about this version of OMIC Age does not include the genetics. It only really includes the first 4. The genetics will be coming soon. And so for those of you interested in what was really sent through the pipeline, we really extracted 61 different clinical phenotypes across 60,000 samples. And so these are 60,000 samples that had been biobanked by the MGB, the Mass General Biobank. And this included demographics, smoking status, other basic variables that are collected, but also including blood urea, nitrogen, potassium, different immune cell calculations like neutrophils, monocytes, eosinophils, etc. We were able to select for specific values that really showed association to time till death or just mortality rate in general. And so by taking the 61 clinical phenotypes, we were able to bring it down to about 27, which showed significant association to overall mortality. And then just to do a little bit more of a data condensation, if you will, or condensing, if you will, we also fit another regression model to really whittle down that 27 to a subgroup of phenotypes that really explained this aging phenotype as well. And this included the Charlson index values, gender, BMI, diastolic, blood pressure, albumin, blood urea, nitrogen, creatinine, glucose, etc. And so using all of this, we were able to develop this metric called Phenobio Age. And essentially what this image shows is that the weightage of all of those 20 features after penalization, what were those associations, or what was the weight of those individual associations or those metrics across. So now that we've been able to create this Phenobio Age, one of the things that we wanted to check was what is that association to the overall chronological age? And what we see is that as this Phenobio Age increases in its value, that is also highly correlated to the increase of overall age itself, with a correlation of about 0.76. I will say on the left is the training, and so we also subjected this to a test model of about, I think it was about 30,000 I think about 10,000 or so individuals, where the correlation was about replicative of the training set as well. And so we've been able to create a value which showed correlation to chronological age, but was still included all of these other clinical values as well. And also all models were adjusted for gender, race, age, BMI, and smoking status. But that association to chronological age is is something to check for, but what is really important is that association to those diseases. And so what we did was we also looked at, and I have to say, Ching Wen and Dr. Jessica Lasky-Sue were very instrumental in a lot of these analyses. So what we're presenting here is also obviously a concerted effort. But when we start to consider the different diseases, we start to see that it actually shows significant associations to all-cause mortality, overall mortality, COPD, depression, stroke, and type 2 diabetes. In both cases, the top is going to be the training, and then the bottom is the testing. So we're able to replicate these significant hazard ratios, suggesting that as the value of this Phenobio Age increases, we start to see that the overall hazard or onset of these diseases also increase as well to a significant value, suggesting that these two values have significant associations. And on top of that, Pheno Age was originally developed as a phenotypic value or a metric as well. And so we actually compared this Phenobio Age to the Pheno Age as well. And what we're starting to see is that the significant associations or the hazard ratios have improved in this bio age compared to the previous the Pheno Age version as well, suggesting that what we're capturing is much more associated to that disease onset, specifically in these disease states, including stroke, type 2 diabetes, COPD, depression, cardiovascular disease, cancer, and all-cause mortality. So we were able to develop this Phenobio Age, but as a company, and especially for the utility of this Phenobio Age, we want to be able to calculate this using just methylation data. And so we went two routes. The first one was, are we able to create a DNA methylation surrogate of this bio age? And to do that, we actually were able to train our methylation data to this Phenobio Age itself. And so to do that, we did a lasso penalized regression model, especially at an alpha 0.1, to predict each not significant protein. In this case, it was just for CPGs itself. And so we were training to identify those CPGs that could actually predict bioage, and we adjusted it based on sex and age as well. And so some of the testing stats that we're starting to see is that what we're able to do, this is the training and this is the test set on the right. What we're able to see is that we're able to create a DNA methylation surrogate which shows a significant correlation between the bioage and bioage itself, even though the error rate is relatively higher. Again, I will show the comparison because this is very important to look at the error rate compared to the OMIC age error rate as well. But ultimately, we're able to see that the DNA methylation version is highly correlated to the clinical version of bioage. But we are starting to see some interesting trends where, especially at higher levels of that Phenobioage, your prediction of that DNA methylation version is actually much lower versus the opposite is happening here at younger ages. And so that was a direct development of the DNA methylation version. However, we wanted to go the Multiomics route to see if imparting more additional omic values, how that would improve upon the overall estimation of that bioage. And so to do that, we actually wanted to go the route of developing surrogate markers from methylation to predict these other data sets. For example, we were able to collect proteomic data, the same clinical value data and metabolite data, and we actually had matched individuals that had all of these data sets as well. And so we pre-processed all these omic data, and what we tried to do was, using those matched data sets, we developed methylation risk scores or methylation surrogate values for each of those proteins. But initially, because we had 27,000 protein groups and almost 8,500 metabolites, we didn't want to create initially. We didn't start with just creating methylation risk scores across all of the panel. We wanted to see which one of those that showed a correlation to this bioage, given that this Phenobioage showed significant association to diseases. So we actually identified those protein, metabolites, and clinical values, which showed correlation to that Phenobioage, and identified about 110 proteins, 25 clinical values, and 299 metabolites that had that association to this aging biomarker. or this aging metric. And then of those, we actually were able to train to the methylation data itself to develop those methylation surrogate models, essentially. And going through the training, the lasso penalized regression, we were able to condense this down just a little bit, which was surprising, to about 107 proteins, 21 clinical values, and 267 metabolites, which equated to about 395 models or methylation models that we could predict each of these values. And again, one of the things that was actually a little bit different here is that we actually ran it based on adjustment of sex and age and no adjustment. And we essentially identified those that showed the highest correlation between the measured value and the observed value based on each of those platforms. And so these next few slides, essentially what I hope you can focus on are really what are the ones that were being brought out? Because one of the things that I hope you can appreciate, and I'll walk you through, is that a lot of these proteins that we identified showed a correlation between the observed values from each of the platforms to the predicted. Those Pearson correlations showed about 0.35 or higher in these models. In addition, we had about 250 or so that showed greater than 0.7 correlation across the board. And some of these are previously created models, but we've greatly improved on, especially considering CRP, which is located right here, which has greater than 0.7 in Pearson correlation. But in addition, we also have FVC and BMI, hemoglobin, different versions of PON1. The reason why there's multiple is because we're considering different protein groups as per the SEER platform. But in addition, we are able to measure additional clinical MRS scores as well, such as FEV1, smoking pack years, BMI, and these actually have about 0.7 or higher in that correlation between the observed and the predicted values based on just methylation itself. One thing to note also is that these clinical values, we have the clinical version of CRP and also the protein version of it as well. So we're considering two different aspects of these individual methylation risk scores as well, which we assumed and which we've also seen improves upon the overall model as well. And lastly, the 267 metabolites, it's the same story. We have many of them that actually are above a 0.7 correlation, and some of these, and overall, we have metabolites that have recently gained a lot of news, such as pyrimidine, which we are able to calculate with greater than 0.2 correlation with great accuracy. And just to reiterate, so for example, when we compare this to what GrimAge was using before, GrimAge was able to use identify about 12 different MRS biomarkers. We have about 396 total, or 395, apologies, and about 353 of those have a Pearson correlation greater than 0.35, and 32 with greater than 0.7 across the board. And this includes, for example, CRP and A1C as well. So with all of that said, we were able to generate these methylation surrogate markers. Now let's tie it all back in if we consider the original figure as well. So developing OMIC MAge, consider the proteomics, clinical, metabolites, and methylation, but you have to consider the rationale. And so the rationale for why we did it this way is because since the initial 2013 clocks, we've also been noticing a lot of issues, especially considering reading the true biological signal, the precision, which means the test and retest. How often are you going to get the same reading if you test the same sample? And then also variations with immune changes. And so this has already been worked on, especially by Dr. Higgins Chen and also with Duneen Pace, where they've been able to develop these metrics that have high reproducibility even upon the same exact sample. And also that reproducibility, or that new computational solution, actually does not change the outcome or association to that disease itself, especially which was greatly described in this paper by Jessica Fall, which was published back in February 21, 2023, which I highly recommend reading. And then another aspect is when we're considering the samples that we test, the cell heterogeneity is also impacting how these overall clocks are being calculated. For example, cell proportion changes our overall biological age score, and we know that certain immune cell subsets have contributed to those very widely varying overall outputs as well. And so this is a bad thing because we don't want these epigenetic clocks to be elevated due to just someone having higher B cells due to a mild illness or just naturally having higher B cells as well. And so we want to make sure that we're measuring aging and not the immune changes that are unassociated with aging. And also on top of that, considering aging actually allows for the improvement or development of novel epigenetic clocks, which are capturing more signals as well. And so I have to give a shout out to Alan Tomasek and the rest of the individuals of the Eric Verdeen group, especially out of the Buck Institute, where they've actually been able to say or show that naive T cells consistently showed a significantly younger epigenetic age than those CD8 positive subsets. And this result suggests that epigenetic clock measurements are affected by those individual cell types, specifically in this case, the CD8 positive T cell differentiation. So again, it goes back to this idea that the cell heterogeneity of the sample will also greatly affect that epigenetic age. And so being able to include that is very important into the model itself. But in addition, by integrating a lot of those immune cell information, you're also creating a model that is highly correlated with additional biological facets that are important or are associated with aging. For example, in Trin clock, by incorporating a lot of those immune cell subsets, showed higher correlation with cancer and also senescence. And especially with senescence, a paper by Kenneth Raj and Steve Horvath, a lot of these current clocks are having a very difficult time showing age change or epigenetic age changes based on senescence itself. And so by incorporating this information, what they were able to show is that this in-Trin clock actually, by taking into consideration a lot of that immune cell components, they're able to be sensitive enough to show changes associated with senescence as well. And so what we actually did was previously we developed a 12-cell immune deconvolution method in collaboration with Andrew Teschendorf and Jessica Lasky-Sue's groups. And we were able to utilize this as a way to predict the overall levels of those immune cell, essentially immune cell levels. And so what it allows us to do is that using whole blood, we can calculate how much of a percentage of those 12 cells of those subset cells, and be able to incorporate that back into the models just so we can consider those fluctuations in those single cells that are part of that whole blood tissue as well. And what was really nice to see from our perspective is that those 12-cell deconvolution, those predictions are extremely consistent with some of those gold standards, such as flow cytometry that were quantified using flow cytometry. And so I'm showing only about 10 of those 12, but we've been able to capture T regulatory, CD4T, CD naive and memory, CD8 cells, B memory, natural killer, monocytes, neutrophils, and eosinophils as well. Now, with all of that background, why is all this important? It's because we actually trained all of these facets, the methylation data, the MRS scores that we developed, and the 12-cell immune deconvolution, along with age and chronological age. We trained that to that bioage that we already know has significant associations to disease. And so here we actually did a 2-4. So we used a penalized model where we're selecting for features that are significantly associated, correlative to bioage, and an unpenalized model where we keep all of this information and train that against bioage itself. And what this kind of this output really shows is that by incorporating all of this information, again, the correlation actually improved between when we started to include all this information. For example, in the penalized model, the correlation between the phenobio-age on the x-axis and the OMIC age or the OMIC-M age is 0.91, versus for the unpenalized, it's a little bit less just because of more information being considered there. But what is important here is, and I'll let you know what the other value was, is that when we consider The penalized model, the mean absolute error, which was originally about 8.7 or so, by incorporating all this information, that error between capturing Phenobio Age and that OMIC age actually dropped to about 4.97, meaning by including additional information, you're actually getting a close enough prediction of that Phenobio Age just from the methylation data itself. Using the unpenalized model, that mean absolute error is a little bit worse than what is there for the penalized, but still far superior than what was there when you're just using DNA methylation itself. And here's a comparison between what happens when you use all of the MRS scores, the metabolites, the CPGs, and immunity convolution, and then when you start to actually select for specific features. What is interesting out of this, and something that internally we're looking into a little bit more, is that once you use the penalized regression model to BioAge, all of the 12 immune cell subsets actually drop out of the model, suggesting that they're maybe not allowing for a good prediction of Phenobio Age. But this is something that we're still considering as we move forward. And on top of that, one of the things that we always think about is that test retest reliability based on those ICC values. And so what we are also seeing is that in the penalized model, that ICC, that test retest, is actually 0.9975, and also in the unpenalized version 0.995. Essentially, what this is suggesting is that when using OMIC MH, the values that come from testing the same sample twice is not differing by it's differing less than 0.003% or 0.003 or 0.3%. And this is on the same level as what we are seeing with especially the Dunedin pace and a lot of those PC clocks as well. And last but not least, the association to a lot of these other diseases. So far, we've been able to test OMIC age on all-cause mortality, stroke, type 2 diabetes, and COPD. And we also compared this to a lot of those PC clocks, including PC Grim age. And what we're starting to parse through but we're happy to see is that it shows high sensitivity to a lot of these disease outcomes. For example, the hazard ratios are higher in both the DNA methylation version of BioAge and the OMIC age as well, relative to PC Grim age essentially across the board. So this was something that we're really excited about. One of the things, though, is that we are seeing some discrepancies, especially with the COPD, as we move forward and as we start to consider additional avenues of future work. This may change as well. And just as an example, this slide is just to show a little bit more of what were the associated weights between the features that were selected for in the penalization. And so what was really interesting to me was the selection of things such as liver albumin, hemoglobin, HbA1c, creatinine, but then also especially looking back at the metabolites and proteins. For example, BMP, bone morphogenic protein 1, was one of those that were selected as well. And so as we move forward, especially with understanding OMIC age, these are some of those MRS scores, or the clinical values, the proteins, and the metabolites that we're going to be researching a little bit more about as we move forward. So conclusions. We developed Phenobio Age, a composite metric of health developed using clinical values and trained to time to death. And what is important about this is that it showed improved associations to all of these different disease outcomes, and especially when we start to compare it to something like PhenoAge, which has been doing remarkable wonders currently. We also used untargeted proteomics and targeted metabolomics to identify 395 different methylation models across those clinical protein and metabolite markers. And a lot of these models show greater than 0.2 correlation between the predicted and observed, and those correlations are improving upon what is currently out there, especially when you consider A1C and CRP measurements. And lastly, we developed a composite OMIC MAH metric that was developed by training methylation, MRS values, immune cell subsets, and demographic information. And it shows improved associations to all-cause mortality, stroke, type 2 diabetes, to PC Grim age, whereas other diseases were still improved upon in previous versions of the clocks. And on top of that, the test retest of this OMIC MH is 0.997, especially with that penalized model. So it provides to be a highly reproducible metric that we are hoping would be very helpful to the clinicians and user, the patients in general. But future work really is that we're going to do a lot more further testing of mortality and disease association, predictability, and test retest on independent data sets. A lot of what we've done is we're using the Brigham's data set that we've been able to develop. So this includes the Nurses Health Study, Women's Health Initiative, and also the Framingham Heart Surgery cohort. We also want to develop more MRS models without using the Phenobio Age associations. Remember, we have about 27,000 proteins and over 8,500 metabolites. So creating those models without actually filtering based on bioage might produce, we think that not only will it produce predictors that are very important for us to understand aging altogether, but may also improve upon OMIC age as well. But also we're training OMIC age to the surrogate Phenobio age. So why not just train it to time till death directly? And that is actually something that we are doing currently. And so one of the questions that really comes up there is, how does it compare between training to this surrogate bioage, Phenobio age, versus just time till death? Is it diluting biological age prediction? I say probably not, but it's something that we still need to test. And ultimately, it goes back to this beautiful review by Justice and Krzyzewski, where it really, in order to make DNA methylation biomarkers into something that's clinically useful, we really have to consider these four metrics: the feasibility to use in clinical trials, robustly associated with chronological age in independent cohorts, predicting age-related change in function, chronic disease, or death, and also, is it responsive to interventions? And so as we move forward, that last bit is especially something that we're going to be considering moving forward. With all that said, thank you so much. I know I hopefully did not go over time too much, but yes, connect with us. Here's social and also our website. If you want to connect with me directly, my email is here, including my socials. And really a big shout out to everyone who's part of this initiative, and also to Qingwen Chen and Dr. Jessica Lasky-Su for being amazing collaborators. And if we have time, I'll be more than happy to take some questions.
Wonderful. Thank you so much. This was fantastic. We have a lot of questions. Are you ready to take them?
Sure, yeah.
Okay, cool. In the queue, first one up, we have Steve.
The original question that I asked way back at the beginning was about the cell populations used to screen the DNA methylation, with the idea that this would be highly variable between different cell populations that would have, for example, a differential response to inflammatory systems like immune cells being in charge of that inflammatory response, and for example, Bugle cells that could be scraped quite easily without it being necessarily inflammatory, with the idea that my recent hypotheses about pathologies, underlying toxic pathologies, is that the pathologies of Alzheimer's disease that are taking place on one side of the blood-brain barrier are comparable in mechanism to the pathologies dealing with COPD and heart disease on the other side of the blood-brain barrier, and that this would have a dramatic influence on your data depending upon what cell population you're sampling.
Yeah. So, okay. So if I'm understanding the question correctly, it's essentially looking at the cell populations and comparing them to, let's say, something that's, for example, Bugle, which is, I guess, less or more resistant to those inflammation changes. Am I understanding that?
It's the idea of what you're looking at. And for example, telomere length depends upon the cells that you're sampling.
So one of the reasons why we really focus on blood versus Bugle, and why and obviously there's always pros and cons, is because, especially when we consider the Bugle collection, especially when we start to go towards more of a DTC or even clinical market, depending on what time of day or even what the individual has eaten or all of those other things, that can really influence how what you're collecting essentially. Now with blood, what we think is that you're mitigating a lot of those issues based on the outside influence, like just outside of let's say just skin, if you will. And so while, yes, with blood, you still have that effect of inflammation and other things, we also think that with Bugle, you're getting a huge range of values that are a huge test and retest becomes a little bit more of an issue, especially when you start to consider that you might be calculating or you might be considering particles that are foreign due to just consumption of food or even just the breathing and taking in additional cell types. And so just for a cleaner, hopefully cleaner output, we consider blood versus the Bugle, even though, as you stated, there are cons with just looking at blood itself.
Have you looked at subpopulations of blood cells as to their data consistency with your model? For example, the aging parameters for inflamed activated cells can be different from ones that are naive. There may be other populations of white blood cells and subpopulations that might show differential response depending upon whether maybe IgE versus IgA or IgM antibodies are involved in those particular populations.
No, absolutely. And I think that's one analysis that we can do, especially given our immune cell deconvolution method, or even just looking at our biobank samples. And so I think that's absolutely something that we can start to show, especially because there was a paper by Jonkman et al., which I hopefully cited, where they showed that natural killer cells actually influence the output of those early epigenetic clocks as well. And so this is something that we want to also show. And I think that we actually have data for which I didn't show, where we do see certain cell populations influencing the overall epigenetic clocks. I'm not saying which ones because I'm trying to remember, but that is something that we have data for, and we can share it, especially when we have this preprint out. But that is something that we'll look into, especially with OMIC MH. But also another thing about the Bugle aspect is that we haven't really been able to identify any validation data sets for Bugle, and the biobank stores for Bugle cells have been very limited as well. And so especially utilizing those blood biobanks have been very crucial, especially to get those additional metadata of the disease states and just overall health metrics, which were instrumental in developing OMIC Age. But thank you for your question. That is something to think about for sure.
All right, we have 3 more. Let's see how many we'll get to. And next one up, we have Cosmo.
Hey, great talk. I think
Thank you.
a couple of us in the chat noticed that there was 2 different clusters of subjects in your scatter plot. Have you guys figured out what those clusters are?
Oh, are you talking about the bio age?
Maybe.
Oh yeah, no problem.
Yeah, it was the first graph that showed a nicely overplotted scatter plot with a single line, but 2 clusters.
That's actually something that's part of the population dynamics that we're looking at. So there does seem to be two different clusters. And right now, the question that we have for BioAge, or why that's the case, we're still trying to figure out. So very astute observation. We'll have an answer soon.
Okay, wonderful. Next one up, we have Tomo.
So I guess my first half of my questions were already answered, but I guess the next one is basically, these will tell you kind of like how long you'll live unless you die a violent and unfortunate death. But people probably want to also learn what they can do about it, and how much can these models be adapted to simpler animal models where you can test interventions like calorie cycling, phytonutrients, and other things, and then kind of do a more targeted clinical study where you go back and say, okay, we're going to choose these 3 things in human populations.
Sure. As for animal models, I think the same mainframe, or like the same kind of, I guess, process can be maybe adapted to, but obviously we're using human tissue, so we are just going to be focusing on humans. But this is where the clinical interventions really come into play, because when we start to consider how these clocks are working, the best way we can do is start to look into clinical trials where individuals maybe are undergoing a caloric restriction. And then from that, how does the overall composite score change? But then also, more importantly, how do those MRS scores that are those individual metrics of the overall composite, how did those change? And so by looking at that, that's one way that we could start to say, okay, as this individual overall is getting younger, it seems like the MRS score for CRP is greatly affected. So figure out ways that maybe you can either increase or decrease that CRP level. And then that could be a predictive method of improving your overall OMIC age clock, if you will. And again, this is one biomarker that is trying to take into consideration multiple, but there are additional biomarkers that we can also integrate. So I think to answer your question, we'll know more once we start to utilize this in those clinical interventions and start to understand, okay, this clinical intervention is overall improving the health of the individual, but maybe OMIC age is not sensitive enough, or OMIC age is drastically reducing based off of this caloric restriction intervention or the application of this other supplement. So that's one way. Those are ways that we can maybe start to test a lot of this clock and honestly any other clock that's developed.
Okay, thank you.
Well, Kuyan, you're up next.
Okay, thank you. But I'm sorry if this was already discussed. What about kind of looking away from all these omics and all this giant data and looking at some really simple clocks like height to waist ratio or grip strength? Do you have any thoughts on that? And is that part of your research program?
So we, to kind of borrow from Google, we stand on the shoulders of giants, right? What we're trying to do is we're trying to develop this molecular based method of quantifying age. But I do think that in the future, we want to be able to compare this clock to those that are already existing, such as height weight ratio and additional clocks. I do think that more recently, these values, especially these lab values that have been traditionally taken from, let's say, blood plasma, they have historically, I think there was a paper, Cannoli et al. 2021, which really showed that these plasma CRP values don't show significant association with age in general, but these DNA methylation CRP values do. And in that same respect, it might be that these molecular age clocks may be showing something that the height weight clocks may not be showing. And so all we're doing is we're adding another facet of this age estimation compared to what has already been there. And I think that that's something we don't want to. This is not a replacement, but but this will add upon our understanding of that aging process in general. But also another thing too is that when we start to consider their associations to disease, the hazard ratios of, especially with these simple clocks, they don't achieve the same level of significance or sensitivity relative to the OMIC age clock itself. Or sorry, not even, let's take OMIC age as one facet, but any of these other molecular clocks as well. And so that's something to start to consider, is that you're capturing something a little bit deeper, especially with these molecular clocks, that maybe is not captured at the same level of sensitivity, especially with these older clocks as well.
Fair enough. Two quick follow-ups. One is, rather than using linear correlation or Pearson rank, how about using mutual information? Because let's take the simple example of height to waist ratio. That will be a U-shaped curve. If people get too skinny, they're not healthy either, as opposed to, so like too skinny and too fat, and then there'll be an optimum, but you won't find that with a linear correlation because it'll show up zero.
No, fair enough. And actually, we've used mutual information as a way to pre-filter CPGs, because we had an entire chronological age clock that we developed. I think it's a fantastic method. Obviously, this is our first run at this, but one of the things that I don't know if Dr. Horvath is on. I don't know why it would be, but I spoke to Dr. Horvath about this, and one of the things that, especially when we start to consider AI and these penalized regression models, these linear models. One thing that he said that really stuck with me is that it's easier to explain what drives a linear model than an AI model. And for us, I mean, we want to understand what are those biomarkers that these linear equations capturing. And it seems to be much easier, especially with those penalized regression models. And by capturing those, we can actually understand more of the biology versus just understanding that we've developed an AI model that has all of these different epochs and different layers, but we can't really explain those layers, like what composes those layers. And I imagine that in the near future, we can go about that. But I think as a first step, really understanding those linear models, I think that makes that allows us to understand more of the biology, and then we can maybe move into utilizing those.
But high mutual information is very simple. It's not a giant multi-layer model. It's just a simple scalar value describing the generalized correlations in a multivariate data set. And so I think that while I agree with what you said, it's a separate issue from a simple mutual information metric, which can capture a little more of a complex relationship, but still be completely explainable.
Sure. No, in that respect, I definitely agree. And I think that'll also come with just trying that method as well.
Okay, we are 2 minutes already over time, which is something we very rarely do. And we haven't even gotten to, there's still 2 questions outstanding. So I will take that to emails. Feel free to email me them and I'll shuffle them to you, or Varun, feel free to drop your email in here. My last question would be, as a closing, what can people in this group do to help you and your work?
I think exactly what just happened these last 5 minutes. Ask if you can ask us questions and ask us your thoughts, and also just ways of improving upon a lot of these clocks. We are more than happy to hear them, and we take a lot of these questions and comments in a way to develop these tools. And so, yeah, I think keeping a supportive and yet critical yet supportive lens is always very helpful. And so, yeah, and one of the reasons why I joined as a fellow here.
All right, that we can definitely provide. Thank you so much, everyone, for joining. Really, really, really enjoyed this. And I'll see you all at the next one. Thanks, everyone, for staying over. I think the 3 minutes really were worth it. So thanks, everyone. See you at the next one.